Assalamualaikum viewers. Please like, share and subscribe this channel. Links of PDF files used in this video are given in description. So today is the part 2 of a lecture series about flow injection analysis, and in this lecture we will see how different components of a flow injection system make the whole analysis. As we seen in the previous lecture that how a typical flow injection analyzer works and what it consists of. So it mainly consists of peristaltic pump and the injector, the transport system consisting of mixing and reaction coils, and then the detector. And this particular setup has actually two channels, which is the carrier stream and a reagent line. Now in this particular lecture we will discuss these different components, and first of all we will see how this propelling unit works. So the propelling unit moves the carrier stream through a flow injection analyzer. Although several types of propelling units have been used, but the most common is the peristaltic pump. As this has been shown, this is a peristaltic pump. So a peristaltic pump consists of set of rollers which are attached outside of a rotating drum. So this drum is rotating and the hollow rollers are attached with it. Now tubing from the reagent reservoir fits between the rollers and a fixed plate. So this is the fixed plate and this is the tubing which consists of the different reagent streams. So as the roller moves, the tubing is squeezed and the peristaltic pump as rotates, then the roller squeezes the tubing, forces the contents of the tubing to move in the direction of rotation. So peristaltic this pump provides a constant flow rate and which is controlled by the drum speed of rotation and the inner diameter of the tubing, and flow rates up to 40 milliliter per minute are possible. However, the limitation of this pump is that it produces a pulse flow, and this pulse flow is more prominent at high flow rates, so that may lead to oscillations in the signal. Second important component in this flow injection analyzer system is this injector, and a typical injector is used to convert about 5 to 200 microliter of the sample into the carrier stream. Although syringe injections through a rubber septum are also possible, but in this particular arrangement, a rotatory or loop injector similar to that used in HPLC has been used. This type of injector provides a reproducible sample volume and is easily adaptable to automation, and we know that automation is very important when high sampling rates are needed. Next important component of flow injection analyzer system is the detector, which gives us the response to the analyte solution. So though most commonly used detectors are actually the electrochemical and optical detectors as those used in HPLC. So these detectors have been designed also around to use ion selective electrodes, and we can also attach an assembly of atomic absorption spectroscopy. So in this way we have a wide range of detectors that can be attached to a particular flow injection analyzer system. Next, the heart of the flow injection analyzer is the transport system, and that brings together the carrier stream, the sample, and any reagents that react with the sample. Each reagent stream is considered as a separate channel, and all the channels must merge before the carrier stream reaches the detector. So, a complete transport system is called as manifold, and the most simplest form of manifold includes a single channel. As in this diagram we have shown a particular arrangement of a single channel manifold. This type of manifold is used for direct analysis that do not require a chemical reaction. In this, the carrier stream serves only as a mean for rapidly and reproducibly transporting the sample to the detector. For example, this manifold design has been used for sample introduction in anatomic absorption spectroscopy, achieving sampling rates as high as 700 samples per hour. A single channel manifold is also used for determining the sample's pH or determining the concentration of metals ions using an ion selective electrode. So here we see that a reagent is coming, the pump is used, which is used for pushing things, and this is the injector, and where the sample is injected. This is the mixing or reaction coil. There takes some time to pass or homogenize things, and then here we have been used atomic absorption spectroscopy or some pH electrode for determining the pH of the solution, or by using ion selective electrode to determine the concentration of metal ions. We can also use a single channel manifold systems in which we monitor the product of a chemical reaction between the sample and a reactant. So in this case, the carrier stream both transport the sample to the detector and reacts with the sample. So because the sample must mix with the carrier stream, a low rate is used. Most flow injection analysis that includes a chemical reaction uses a manifold with two or more channels. So including additional channels provide more control over the mixing of reagents and the interaction between the reagents and the simple sample. Two configurations are possible for a dual channel system. A dual channel manifold, such as shown in figure A, is used when the reagents cannot be pre-mixed because of their reactivity. So if we see here, this is the reagent 1 and this is the reagent 2. Both are pushed together by the peristaltic pump, and these are mixed together just before reacting with the sample. So as sample comes from the injector loop, then this sample is mixed with the product of reagent 1 and reagent 2, and then here the reaction or mixing of these substances happens, and then the overall product is fed to the detector for recording its response. Second type of manifold using a dual channel, we can also use this arrangement, and in this arrangement we commonly add a second reagent after injecting the sample into the carrier stream. This style of manifold is used for quantitative analysis of many analytes, including the determination of wastewater's chemical oxygen demand. So if we see here that first reagent 1 is pushed and it is directly mixed up with our sample, whereas reagent 2 is then reacted or mixed with the product of reagent 1 and the sample. So here the reaction happens, and then we fed this to the detector to record the detector's response. So here we have actually seen two arrangements, and in the first arrangement of dual channel manifold, both the reagents are combined together before mixing with the sample, whereas in the dual channel system or manifold, the reagent 1 is first mixed with the sample, and then reagent 2 is mixed with the sample and reagent 1 product. So here we can do a lot of customization in order to control the mixing or the stages at which a particular reagent reacts. More complex manifold systems involving three or more channels are also commonly used. So one example is a 4-channel manifold as shown in figure 1. Here this is the channel 1, channel 2, channel 3 and channel 4. So what is happening here, that reagent 1 is combining with the sample, and then reagent 2 is combining with the product of reagent 1 and sample 1. Similarly here the reagent 3 is mixed with the product of reagent 1, 2 and the sample, and here in this channel reagent 4 is mixing here with the product of reagent 1, 2, 3 and the sample. So in this way we have a more control over the reactant species, and in this way we fed all this to the detector and record its response. Incorporating a separation module in the flow injection manifold, we can include a separation. The separation of maybe of dialysis like gaseous diffusion and liquid-liquid extractions. Although these separations are never complete, they are reproducible if we can carefully control the experimental conditions. So, as we shown in this figure, here are the mixing and reaction coils. So in order to do separation of the analyte mixture, we will incorporate separation modules in these places. The separations like dialysis and gaseous diffusions are accomplished by placing a semi-permeable membrane between the carrier stream containing the sample and an acceptor system. So as the sample stream passes through the separation module, a portion of the those species capable of crossing the semi-permeable membrane do so, entering the acceptor system. This type of separation module is common for the analysis of clinical samples such as serum and urine, where a dialysis membrane separates the analyte from its complex mixture or matrix. Semi-permeable gaseous diffusion membranes have been used for determination of ammonia and carbon dioxide in blood. A typical separation module for a flow injection analysis using a semi-permeable membrane has been shown in figure. The smaller green solutes can pass through the semi-permeable membrane and enter the acceptor stream, but the larger blue dots, which are representing solutes of larger size, these cannot pass this semi-permeable membrane. So although the separation is not complete, note that some of the green solutes remain in the sample stream and exist as waste. So the blue is going towards the waste, whereas the analyte or the particles of our interest are reaching towards the detector. So it is reproducible if we do not change the experimental conditions. So in this way we can incorporate this module, which is using the phenomena of diffusion through semi-permeable membrane in order to separate our analyte from the sample stream. Liquid-liquid extractions are accomplished by merging together two immeasurable fluids, each carried in a separate channel. The result is a segmented flow through the separation module consisting of alternating portions of the two phases. At the outlet of the separation module, the two fluids are separated by taking the advantage of difference in their density. For example, in this particular figure, a configuration of a separation module in which the sample is injected into an aqueous phase and is extracted into a less dense organic phase that passes through the detector. So if we see here, this is the stream of the organic phase and this is the stream of the aqueous phase. Actually our sample is contained in this aqueous phase, but we are mixing these two phases together. So if we see this portion, the white portion, that is the portion which is coming from the organic phase, whereas the blue portion, this is the portion which is coming from the aqueous phase. So both these are combining together. However, what is happening that the analyte is coming from our aqueous phase and is shifting towards the organic phase. So if we see here, the color of this blue dot is changing. What it is representing here, that our analyte is exiting from this phase and the color of this organic phase is periodically changing into a red. What is happening, that analyte is withdrawing itself from this aqueous phase and is entering into the organic phase. Now when the both these things reach together, so here we are using the density. As the density of aqueous phase is greater, so it will be percolate down, whereas the density of organic phase is lesser, so it will stay in the top and this stream is passed to the detector in order to get the signal. So in this way, by inserting this separation module for the flow injection analysis, we can do a liquid-liquid extraction. In the set, there has been shown an equilibrium reaction. As the sample moves through the equilibration zone, the analyte extracts into the organic phase. So in this way, by incorporating the separation modules using the semi-permeable membrane, and by using liquid extractions we can also do separations. So these separations are not complete, but these are reproducible, so which is of great advantage. So this was all about the instrumentation and different components and the working of individual components that are used in a particular flow injection system. So this was all about today's lecture. If you have any questions, you can ask them in the comments. Thank you. Allah Hafiz.